the title of this is Biology from a Molecular Perspective, and I want to really help you guys understand a bit more about how biology works. Because no matter which technologies win, so to speak, for freedom of form, whether it's genetics or bioprinting or neuroprosthetics or even just surgical approaches, it's important for us to be up to speed on the basics of how biology really works. This is all still a learning experience for me too. I don't know everything. A lot of what I'm about to present, I actually had to look up myself because there's just a lot of knowledge out there and there's a lot to distill. But the more that we learn as a community, the more that we understand, the closer that we get to success. And even though a lot of the bleeding edge research is done in research labs, as a community, we still have the responsibility to understand as much as we can and to try to help out researchers wherever we can. So, you know, this isn't necessarily just for fun, even though we can have fun. I think that we have a duty to do our best to understand these things. So with that, I'm going to go ahead and get started. Maybe. Okay. I just tried going full screen here. Does it still look right to you guys?
Yes, it looks quite good.
Excellent. Okay, so starting right off then, I'm sure that we've all been in biology lectures that start off with the essential dogma of molecular biology. It's where DNA is transcribed into RNA, then RNA is translated into protein, and they always go into excruciating detail about what RNA DNA-dependent polymerase is, what a ribosome is, you know, what a Golgi body is, and yeah, mitochondria are the powerhouses of the cell. And, you know, by the end of it, you have memorized a list of stuff, and I feel like you don't really actually understand anything better about how life works. So that is what I exactly don't want to do in this seminar. And I hope that we can all agree that that is a desirable outcome. So instead, I want to have us focus on the following. I want to help you guys understand how processes inside cells work. And the more of a tangible and realistic feel that you have for it, the better. I want to have you appreciate the complexity of biological systems without losing the forest for the trees. Like, I don't want to go through like a memorizing list of parts. I don't care what you know, alcohol dehydrogenase is, or you know, the set of 40 or 50 Hox genes. Like, I don't care. That's excessive detail, and I'm sure that you don't care about that level of detail as well. Then finally, I want to help you understand the interfaces of how cells communicate, how they work together, and how they ultimately settle on regulatory and other actions to perform, you know, in the process of acting as tissue. So, you know, again, it's more of a focus on the interface and building a intuition for this stuff that I really want to do. And in this talk, I'm dividing it into 3 parts, basically. Are you able to see my mouse movement, by the way? Or does that not show up? Yes, you are.
Yeah,
I can see your
cool.
mouse.
I can see it, yes.
Excellent, that's cool. That is really cool. I am a noob at Zoom. Okay, so the 3 principal parts of this talk are going to be, you know, physically, what are cells? Are they squishy, for example? How they can move, and you know, how that is all accomplished through molecular mechanisms. And I have to confess that I really didn't understand biology until I really understood the cytoskeleton. Like everything just looked like cartoons and diagrams on a page until like a professor actually explained the cytoskeleton well to me. I think that the cytoskeleton is really key to understanding how life works even. Um, the second part of this talk will be about cell memory and decision making. There will be a little bit of small focus on gene expression there. And then finally, the third part will be going into a specific molecular mechanism that is used to accomplish a specific biological function. More on that later, and we are just going to focus now on the physical composition of cells and tissues via the cytoskeleton. So a good thought experiment of what the cytoskeleton does is you can have, like on the left, there is a blob of water, and if you poke it, it squishes. And you know that's all well and good, but if you want a like a organism to be able to move through its environment to not just squish whenever a light breeze hits it, then it needs to be reasonably strong. And this also applies for anything that's mechanically important, from pumping fluids around the body, such as the heart, or even during embryonic development. Anytime something has to move past something else, or anytime something has to resist forces, you need some structure inside cells. And if you poke this thing, which is reinforced with these springs, then you will see that it's just stronger and doesn't squish. So that is, in a nutshell, what the cytoskeleton is accomplishing. To go a little bit more realistic, you can say that the shapes and strength of the cells are a result of tensegrity. They do exert a positive pressure from inside. Like they will tend to bulge outwards because the fluid inside is generally a little bit higher pressure than the fluid outside. And that force is resisted by lines of tension that run roughly from the center of the cell to the periphery. And these tension lines can be arranged in specific ways to influence cell shape. And they can also line up across cells. Like you can see here, I've diagrammed these two tension lines lining up fairly closely. And if you start to assemble cells into large tissues, and if you're more careful with how the cytoskeleton is lined up, then you can start to arrive at some pretty elaborate structures, like cytoskeletons kind of cross-linked across membranes. I believe that's called a focal adhesion, to like kind of cellular superstructures. And that integrates with extracellular polymers, part of the extracellular matrix, to wind up with something that is kind of strong overall. And this is an attempt here to diagram a membrane that's lining a lumen, such as a capillary, or such as a lymphatic duct, or the intestine. It doesn't really matter. The point is, this is just like a membrane that is resisting some source of pressure. So that's like hello person. Hello, thanks for joining. Okay, so that's like in general what the cytoskeleton, along with the extracellular matrix, along with cell-cell adhesion, are accomplishing is that mechanical strength. So that's as if everything was just sitting still. those are static forces, so to speak. But of course, as you know, life moves, and you can use the same molecules to accomplish the movement of cells past each other, as well as the regulated contraction of cells. So in the first example, specific movements in the cytoskeleton on one side of a immune cell makes it able to move through the environment. This is done in combination with the specific adhesion and then release from a surface membrane. And you can easily get down a rabbit hole of detail, but the point is this is a very specific regulatory activity that depends on the cytoskeleton. Conceptually similar, actually, is the movement of what's called a axon growth cone. This is used in embryonic development when you have like brand new baby neurons that are just like basically balls. These neurons, or call them precursor neurons, they're not connected to anything, and in order to function as a neuron, it has to connect with something. So the growth cone is the way that they extend their axon out to connect with the target. And it's actually kind of similar in mechanism to immune cells. It relies on the outreach of little fingers or lamellipodia that respond to specific directional cues, then ultimately drag the axon behind them as they grow towards the target. Then finally, as like a third example of the cytoskeleton in motion, we have muscle contraction, obviously. You've got like microtubules, and then molecular motors, and in response to a cue, the motors will tend to move past the cytoskeleton and result in a contraction of the overall muscle fiber. And this is kind of telling that I can't remember what I think that this is myosin that does it, but like there's dynin and kinesin and like who cares? There's just like a bunch of motors, and I feel like this is another place where it's possible to get lost in detail. Like there's so many motors and so many different varieties of microtubules that you can easily get lost. But the point is the overall concept relies on the cytoskeleton being used to accomplish a motion. And that's why I really want to get across, is that you can do a lot with just active movement of the cytoskeleton. Come on. There we are. Okay, so we can now put a little bit more specific names on this. This is a zoomed in picture of a cell, or maybe call it a cartoon of a cell. Here I'm showing microtubules are extending out from the center of the cell towards the surface. These are often cross-linked with actin. There's like a specific molecular complex here that's able to do it. I forget what it's called, but through some indirect interactions, microtubules can bind actin. Actin then attaches to integrin, and integrin attaches to the extracellular matrix. In addition, you can also have cadherins linked from one cell to another. So like you can see that, well, there's like a variety of different microtubule types. There's a variety of actin types. There's like several integrins and several cadherins, and several varieties of extracellular matrix. But the point is, these general mechanisms ultimately make it possible for these cells to both be strong and to move relative to each other and relative to the environment. So that is that. And now moving past cartoons, This is just to show that this isn't just like made up junk on a page. I think that this is actually really important to understanding how the stuff actually works. Is giving like 2 actual examples of what the cytoskeleton looks like. So on the left, we have, I just grabbed this picture off the internet. I think this is a fibroblast because like everything is fibroblast, but like here they have stained the microtubules in green. You see them kind of extending towards the periphery. Then they've got actin generally towards the surface. And because this cell is not moving, you can see that there is a very even distribution of proteins throughout. Like actin is on the right side, it's on the left side. The microtubules extend in basically all directions. And, that is a very, say, non-polar way of laying out the cell. In comparison, we've got the growth cone of a neuron here on the right, and this, you can see that this is very different looking. The microtubules are lined up in parallel going down the shaft of the growing axon. Then the actin is very well aligned, extending out radially throughout the growth cone. And these individual spikes are all feeling and sensing the environment. And whichever side, like over here, maybe the left is expressing a specific cue, the actin will ultimately grow more in that direction, steer the growth cone. Then these extensions of actin over here will break down. So this is like a very dynamic, rapidly changing process, and it relies on this very well-structured setup of actin and microtubules. So I think that this is actually really cool and awesome, and really makes it clear that this cytoskeleton is key to making cells do whatever they do. Let's see, I think that's the last. Oh yeah, there's one more point here that I want to cover in this section, and that is what cell-cell adhesion can look like. Let's see. These are, if I remember, epithelial cells. This is just another picture that I grabbed off the internet, but you can see that stained brightly here is a bunch of cadherin molecules. On the left and right side are both cells. They are dark because they're just not stained with anything. And the boundary of one cell attaching to the other is just dependent on these cadherin molecules, which you can only see because they've specifically stained cadherin. Okay, so that is actually the first third of this talk. We've gone through what cells and tissues are physically. We found that they are squishy, but maybe not as squishy as they would be without a cytoskeleton. We found that the cytoskeleton helps them move, and we've at least named a few specific parts of different molecular categories that enable cells to physically interact with each other in the environment. So before we go on, I just want to ask, like, are there any questions or comments? Because like I feel like I've been just talking at you for, geez, what time is it? Yeah, like 20 straight minutes. Any questions? Hmm.
It's been a good clarification from because I haven't been in like a biology class since like over a quarter of a century, so it's a pretty good refresher and clarification. So I probably don't have much feedback.
Okay. Going forward, if anyone has any questions, definitely feel free to. So yeah, I will go ahead and keep moving. The second part of this will be about cell memory and decision making. I'll talk briefly about gene expression, what it is, what it looks like. And then I'll go through how context, that is like a cell's environment, can influence its decisions in a molecular way. So, without further ado, let us go on. So first of all, I actually literally just said this. So one significant statement is like, cell memory, cell decision making. The ultimate consequence of this is that different types of cells do things differently. Of course, a neuron should be different, consider different information than heart cells or skin cells. So cell memory and decision making is basically a fancy way of saying just that. It's actually pretty simple in terms of importance, and cells have clever ways of accomplishing that. So let's have a hypothetical cell type A and cell type B. They have a identity, which here I'm just going to say is the types and amounts of molecules that are produced. I think that this is the easiest to verify in terms of what a cell identity is. There are other ways, like shape. Like a cell type A might have a different shape than a cell type B. But the thing is, a molecular identity is easier to quantify, and it's easier to get down to the mechanism. So that is why I really want to drill in that molecules are really important to establishing the cell identity. The cell identity, the types of molecules that are produced, is ultimately a product of gene expression. To talk a little bit more about what this can mean for these two different cell types. In cell type A, I've shown a few green diamond molecules. Then cell type B, I've shown a lot more blue square molecules. Maybe these are like evolutionarily similar molecules, or maybe they're totally different. It doesn't matter. The point is they're expressing two genes that are non-overlapping. In comparison, maybe cell type A is expressing this orange ball. Maybe it's expressing this orange ball a lot. And then maybe cell type B is expressing this orange ball a little bit. This is a quantitative difference in gene expression. Cells are able to do this through careful regulatory mechanisms, like just the amount of a gene that's produced can influence cell identity. It doesn't have to be black and white. Another aspect of gene expression is you can have surface proteins. Surface proteins might, this is a pretty standard way of showing it, by the way, is you can have like a purple thing that's pointy. You can have a magenta thing that's pointy in an opposite way. You can have like basically a hoop. This implies that cell type A and cell type B might fit together in some way. And the hoop, implies that you might have some other molecule floating around then landing in there. So this is just a descriptive overview of how cells can differ based on gene expression. So, what is gene expression really? Well, as a reminder, we have the central dogma. This is basically just a rebranding. The central dogma says that you have DNA that is expressed as RNA. Thank you. That ultimately reaches the state of being protein. Now, granted, some RNA is never turned into protein. That's just called non-coding RNA. And RNA can sometimes act as an enzyme to have like active metabolic activities. So it doesn't always get translated into protein. But the point of this is that gene expression arises from DNA, and you can look at gene expression either at RNA or at protein. Both are consequences of this gene in DNA being expressed. And that is ultimately all that I mean. So, that's like a cartoon. And now on this slide, I want to get across the impression that there is actually a lot of detail here. On the left, we have cell type specific genes. And the red just means that there's a lot of a gene being expressed. Then the blue means that there's not much of that gene being expressed. So for example, we can say that our cell type A from before has a lot of these red genes. Then our cell type B from before might have a lot of these genes later on. So, moving from like that cartoon to real life, you immediately see a little bit more complexity, of course. But the principles are still the same. And that, I think, is really important and makes things a lot more conceptually easy, is just, this is just a lot more things that all use the same mechanism. So to put some specific names on this, on the left, we have some genes involved in astroglia. Then in the middle, we have a list of genes that's used in neurons. Then finally, we have a list of genes that's used in oligodendrocyte cells. These and astroglia are just both types of glia. Details not too important, but there's a lot of genes that distinguish these cell types. And those genes basically form the basis of cell identity. Of course, this is life, it's messy. So on the right here, there is another diagram of gene expression which is not so clearly distinct between cell types. I'm not going to go to details here, but there's like, on the x-axis here, there's like different cell types, and you can see going up that these different cell types have very overlapping gene expression. So we've got astrocytes and then neurons all expressing this set of genes. Again, detail's not important. The point is very different cell types can share expression for some genes. And that is all that I want to say about that, because this can quickly become a rabbit hole as well. Taking this back out into real life in terms of what it looks like in tissues, this is a pretty standard way of visualizing gene expression. Like having a graph of red and blue is great. It's quantitative, but that doesn't tell you what it actually looks like in the tissue. This is what it looks like in the tissue. So in panel 8 here, we've got a cross-section of a mouse brain. Give you a sense of scale, this is just 1 centimeter across. And on the surface here is the cortex of the brain. The purple staining indicates gene expression of whatever NOV is. Well, first of all, we're in December now, not November, so this is probably not standing for November. I don't know what it stands for, and it doesn't really care either. But the point is, like some cells, basically everything here is a cell. Some cells express it a lot, some cells don't express it at all, and some cells express it a little bit. Then going on, we have like this other cell type, these astrocytes. They're expressing this other gene in NTSR2 in a pattern that doesn't quite match up with the neuron pattern, as you'd expect, because they're different genes and different cell types. And then finally, we have this other cell type, the oligodendrocyte, expressing yet another gene in a different pattern. All this is to say, again, that you can take all of these, going back a slide, that you can take all of these like graphs of red and green, red and blue gene expression, and then put them in real tissue. And this helps you start to understand how the entire organ works, because you can see, oh, this astrocyte is like kind of close to, these neurons over here, and maybe they interact in a specific way. So that is pretty much that. So one other thing that I want to mention about this is that you can do this for like all 20 or 30,000 genes that exist in the genome. Obviously, that is a lot and too much for anyone to go through. However, there is a massive database of gene expression, and you can go there at mouse.brainmap.org, or other organisms at just brainmap.org. And you can start to really appreciate like what this massive amount of complexity and diversity looks like when applied to real tissue. It's actually really interesting, and I've used this a lot, and I really encourage it because it's just really cool to look at. And it's programmed well as well. It's nice to browse through. It is not your traditional wreck of a website that normally comes up in science. It's actually really well done. But anyway, that's an aside. Let's see. Right. So that is gene expression. So now we can move on to context. Expressing genes is one thing, but that doesn't really inform you how cells can work together, how cells can consider their environment to make complex decisions. So as a example for this, I'm showing two different situations, situation A and situation B, which I'll get to shortly. In situation A, I have this diffusible molecule, this red ball, that binds to this receptor. And I've illustrated here that there is nothing on the other side of this receptor. That means that really this receptor is not able to cause a signaling cascade because it's not connected to anything. So what this results in is, step one, the molecule lands on the receptor. Step two, nothing happens because the receptor isn't connected to anything. In the situation B, I'm now showing a binding event between this cell and this cell using this cell-cell receptor molecule. And this sort of stuff happens all the time. So basically, what this binding event does is it lets this cell know that it's now near a neighbor. And conceptually, that's how it's thinking about it is, oh, I'm near this neighbor. Mechanistically, this binding event from this receptor, okay, thank you, will cause more of these other molecules to bounce around the cell, then bind to the other receptor. So now I'm illustrating these green diamonds binding this receptor, these green diamonds binding that receptor, and then these green diamonds binding this receptor. So you can now say that this receptor is primed in a way to induce a signaling cascade. So now, when you have your diffusible molecules hit the receptor, they're able to transduce a signaling cascade because it's made possible by these green diamond molecules. And then to show a schematic of that, the steps are, first of all, you've got the cell-cell binding event, which produces the green molecule. The green molecule primes the receptor. Then when the receptor is bound, the complex is able to induce a signaling cascade that results in some arbitrary activity of that cell. And you can just choose whatever you want. So that is like the basic takeaway from this is that even with one type of receptor, this red receptor, that you can have multiple outcomes depending on what the context is. And the context, such as sensing a different cell in the environment, ultimately comes down to molecular mechanisms. I feel like I'm going to sneeze. Oh God. And it went away. All right, so moving on. So one other way that that context can work, besides what I've just shown here, is you can have this other cell type with a similar but different receptor, and the separate receptor might be able to induce a totally different sort of signaling cascade. So here, the context is just expression of this receptor that differs from the other receptor. Here, it might not rely on another cell being nearby. It might just be call it an intrinsic context. And then ultimately, when that receptor is bound, it might cause cell type C to cause a separate action. So that is like a overview of some types of mechanisms that can be used to influence the outcome of decisions based on the environment. That is basically wrapping up the second part of the talk. We've covered gene expression, including how what is kind of sort of the central dogma can lead to the expression of molecules that influence what cells do and what their identities are. And we've learned about how different contexts can influence decisions and decision outcomes depending on just what molecules are actually expressed. And then, no questions still. I'd like to now go on to the third part of this talk and go through a bit more of a intense example of how molecular mechanisms can talk to each other and just ultimately accomplish a biological function. So I'd say that this example kind of wraps things together. So what does NMJ stand for? It is the neuromuscular junction. And there's, to be fair, a lot of stuff that goes on here. The neuromuscular junction is very complicated, and it does its function basically perfectly. Its function is to bring a signal from a motor neuron, a electrical signal, and then the signal causes the muscle fiber to contract. So this is what makes it possible for your muscles to contract. Simple, but the mechanisms are a little complicated. So first of all, this should be a pretty familiar example of, first of all, just you've got action potentials and muscle fiber contraction results. In the middle here in yellow, I'm showing a axon terminal, and this is called a synaptic bouton in this part. And the axon terminal is surrounded by terminal Schwann cells. These are a type of glia. They support survival of the neuron as well as improve its electrical properties by acting as an insulator. This is all very closely juxtaposed to a specific site on the muscle fiber. So these these are specific receptors that will ultimately capture some molecule expressed from the neuron. The neuron is kind of primed with a few vesicles near the synaptic terminal or near the synaptic cleft, where given a signal, these vesicles will be secreted and release this purple stuff into the synaptic cleft. So let's now imagine that we have an action potential. That action potential causes electrical charge to build up here. Or actually, this is technically a reduction in charge because it's depolarization, but whatever. This electrical charge is picked up by mechanisms that bring these vesicles to the surface of the neuron and then secrete this purple stuff into the synaptic cleft. So the purple stuff ultimately binds these receptors. The purple stuff is acetylcholine, and it binds acetylcholine receptors. These receptors then are they open ion channels and induce what's called a excitatory postsynaptic potential. So charge builds up because of the result of those receptors. And then let me play that again, actually. The charge transmits very quickly through either side of the muscle fiber. Being electricity, it's very easy to go from one place to another. Then it lands in these transverse tubules. Then ultimately this charge here hops over, then binds to something which I didn't draw here called a sarcoplasmic reticulum, and that is ultimately what causes the muscle fiber to contract through a calcium-dependent pathway. Again, like you can get very quickly lost in those details again, but the point is you've got a neuron, glial cells, and a muscle fiber all close to one another in a very carefully arranged structure you have a variety of mechanisms depending on charge, specific localization of molecules, specific organelles, diffusion of molecules, and then a receptor binding that triggers yet another event. So this is all a complex process that depends on simple inputs, charge. And ultimately that causes something that is, you know, physically meaningful in the real world, muscle contraction. So this is I would say, a prime example of how you get from something simple, again, charge, to something that really impacts the real world. And you've got a bunch of cell types, and you've got a bunch of molecular mechanisms in the middle. Right, yes, blah, 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 and I can only animate so much. Sorry. Like this is a interesting process to go into further. It's just maybe a little bit excessive detail for our point right now. So something else about the neuromuscular junction. It's all well and dandy to understand how it works in the adult, but that doesn't really tell you, how it gets to be what it is. And this is also, I would say, important to understanding biology. Everything in biology is always moving. The neuromuscular junction is the result of specific actions, and it takes active effort to maintain as well. If you're familiar with the expression "use it or lose it," and that is still true of muscle fibers. And it takes active regulation to maintain those neuromuscular junctions in the adult. Looking back into the development of the neuromuscular junction, because again, this is important to understanding how biology works. I'm going to say that development of the NMJ depends on cells interacting with each other in specific ways. It depends on cells moving through the environment, and it relies on changes in cell identity over time, because a muscle fiber has not always been a muscle fiber, and that's a pretty interesting process. So I took these figures from a 1999 article in Annual Reviews by Josh Sains and Jeff Littman, and I just love this figure. It really wraps up the ideas very nicely. So from the top here, they are showing a extension of a growth cone, outgrowth of a new neuron, and that's accompanied by Schwann cells. So these are actually migrating at the same time to where they need to be. From the bottom, we've got muscle cell precursors. These are called myoblasts. They don't look anything like a muscle fiber, but they eventually self-arrange, through regulated mechanisms and interacting with each other. They ultimately break down their cell membrane in specific ways and merge into a unified muscle fiber. Then, as the development proceeds, the axon's growth cone, over here in panel C, by the way, the axon's growth cone will contact with the specific site on the muscle fiber, and start forming those synaptic boutons. That's yet another specifically regulated process. And the Schwann cells will help guide development of that niche as well. So, through the careful movement of the cells and changes in cell identity, we ultimately arise at what we call a neuromuscular junction. All right. So another aspect of the neuromuscular junction, which is worth discussing, is linking this back to molecular mechanisms. We've talked about, in part one, we talked about the how it actually functions, which is, dependent on charge and causing contraction. We just discussed how it develops, and now we're going to discuss very quickly like the molecular mechanisms that are responsible for setting up and maintaining the neuromuscular junction. Details here, as with anything else, can very quickly lead you down a rabbit hole. But the point is, these authors have illustrated a Schwann cell, a motor neuron, and the muscle fiber. The Schwann cell actually has to actively signal using a diffusible molecule, and that's picked up by the motor neuron. These other diffusible signals called FGFs are secreted by the muscle fiber, and FGFs are they're a big protein family. Don't really worry about the details, but they are able to diffuse, and then they're picked up by specific receptors also on the motor neuron. Another diffusible factor called GDNF, glial-derived neural factor, who cares? We've also got integrin, which links to laminin. And then we've got collagens as well. So we've got a combination of diffusible factors. We've got cell-cell binding, and we've also got cell extracellular matrix binding, all of which have to work in tandem to guide development and then maintenance of the neuromuscular junction. So this is, as you can imagine, a very complex process that ultimately involves hundreds of genes, and all of them are working in parallel through a variety of complementary mechanisms. So, if you just understand that concept of stuff being secreted by Schwann cells, the muscle fibers, picked up by the motor nerve, then if you have that combined with adhesion and extracellular matrix interactions, then you can start to understand the basics of how cells talk with one another, and how they even have a three-way communication in the process of developing a desired structure. So moving on from here, this is also, I think, helpful for understanding, is just seeing what it actually looks like. This is a electron micrograph, and this whole view is probably just a few microns across, and the resolvable features are a few nanometers across. So in the center here, we see a neuron's synaptic bouton. The SBL is the basal lamina. This is extracellular matrix that is very closely linked with the neuron as well as surrounding cells. Over here, we see all these dotted patterns with the muscle fiber. Those are the aligned units of the cytoskeleton, just cut across and transverse. And then over here, we've got a Schwann cell kind of wrapping around. So we see that the cartoon that I showed before of a Schwann cell, neuron, and motor muscle fiber is actually reality, but the reality is a little bit messier than what you can see. And this is also just really small. And we can also start to appreciate kind of why science is hard, because it takes a lot of effort to be able to observe this. Then the final point that I want to communicate about the neuromuscular junction is that circled in red are mitochondria. And as we know, mitochondria are the powerhouses of the cell. But being silly aside, this is probably one of the best demonstrations that cell organelles do matter, but it's just a lot of detail. So a lot of the time, you can just assume the organelles will find their right place. Mitochondria are actually actively transported to the right place along the cytoskeleton, actually. And a lot of times you don't have to concern yourself with the regulatory mechanisms of mitochondria and other organelles, because a lot of that stuff will just happen automatically based on upstream regulatory activities. So even though mitochondria and other organelles exist, I would say that they're not as essential to understanding how regulatory decisions are made. Sometimes, but mostly not. So to review, because again, the neuromuscular junction as well as this development are pretty complicated. The NMJ performs a muscular function that results in muscle contraction. It relies on specific cells being in the right place. It uses simple signals like electric charge and acetylcholine in specific ways. And you can have electric charge and acetylcholine anywhere else, and they won't work. They only make sense in the context of the neuromuscular junction. The NMJ arises from a self-organizing process that relies on cell-cell interactions, cell movement, and even changes in cell identity over time, because the muscle fiber doesn't always exist. It comes out of the merging of progenitor cells. The NMJ uses a lot of different and complex signaling pathways to guide its development and to maintain it in the adult. The NMJ, as with a lot of interesting things in biology, is very hard to observe directly, and it takes a lot of work. And this, again, is why biological sciences are hard, is because it just takes a lot of effort to observe and assimilate these concepts. Then finally, the NMJ does rely on cell organelles being in the right place. But again, I would say that conceptually, the bigger focus is on how it comes to be, and the proper placement of cell organelles, in my opinion, can be assumed to happen roughly automatically. So that does it for this third and final part of the talk. We don't want to go through that. So that will actually do it. Are there any questions or any comments?
Yeah, I kind of had to dive out of for a few minutes because I've been dealing with some cron crud and blowing my nose and whatnot.
Ooh.
But yeah, it's actually sorry. But yeah, I definitely do want to rewatch this thing again when I can give it my full concentration.
Okay. All right. I'm glad to hear that. Drakar, anything?
Nothing so far.
Okay. All right. Well, in that case, that should do it. Yeah. All right. So in that case, I'll just call it there. Then I'll make sure that this recording is uploaded to the YouTube channel as soon as possible. So yeah, thank you guys for attending. Appreciate it. And I hope